What's up, students? Today we're going to start a biology series. The first chapter we're going to talk about is the cell cycle. This includes interphase, mitosis, and cytokinesis. What are the stages of the cell cycle in detail? The first stage is interphase. This consists of gap 1, S phase, and gap 2. Second is mitosis. This consists of prophase, metaphase, anaphase, and telophase. And the final step is cell division. This is cytokinesis. Here is a graph showing the cell cycle of a cell dividing by mitosis. As you can see, the large proportion of the cell's life is in gap 1. Interphase. What happens during interphase? The cell grows to its normal size following its previous division while carrying out its normal function. The cell then receives a signal indicating that it should divide. The gap between cell division and synthesis phase is gap 1. The gap between synthesis phase and the next cell division is gap 2. Interphase. In gap 1, the cell makes RNA, enzymes, and proteins. In the synthesis phase, the DNA is replicated. Two sister chromatids make up one chromosome, and the construction of kinetochores take place. Gap 2. The cell continues to grow. The DNA made during the previous phase, the S phase, is checked, and errors are repaired. This prevents mutations from occurring. Cell prepares for division by a sharp increase in the protein tubulin. This is needed to make microtubules for the mitotic spindle. Replication of centrosomes in animal cells, as plant cells do not have centrosomes or centrioles. Next step we're going to talk about more in detail is mitosis. What happens during mitosis? Cell growth completely stops. This means that no RNA is produced and no protein is produced. The whole cell divides to create two genetically identical cells. Here are the stages of mitosis: prophase, metaphase, anaphase, and telophase. At the beginning of mitosis, two centrioles are found in the cytoplasm. The two chromatids are connected by a centromere. Each chromatid contains an identical copy of DNA. Each centromere has two attached kinetochores. What's the difference between centrioles, centrosomes, and centromeres? We're going to start with centrosomes. They are microtubule organizing centers, present only in animal cells. The spindle is made out of microtubules. The spindle is needed for the separation of sister chromatids. Centrosomes are a pair of centrioles surrounded by proteins. Next is a centromere. Centromere holds the chromatids together. The centromere is where the spindle attaches, at the kinetochore. Kinetochores are made of proteins. They connect the centromeres to the spindle. As you can see, they are the pink highlighted area. The first stage is in metaphase and the second one is during anaphase. The first stage is prophase. It's divided into two phases: early prophase and late prophase. Prophase I. The nucleus and nucleolus are present, visible by staining. The chromosomes have two telomeres, one at each end. Chromatids form long strands. They are not coiled up. Two centrioles are close together, next to the nucleus, and are perpendicular to one another. The spindles have not yet formed. Next up is late prophase. The nucleus and nucleolus are absent. They are said to dissolve. The chromosomes have 4 telomeres. They are doubled. Centrioles start to move to opposite poles. A spindle starts to form. The mitotic spindle is labeled on the diagram. The second stage is metaphase. In metaphase, each centrosome reaches a pole. Centrosomes organize the spindle production out of microtubules. Chromosomes line up across the center or the middle of the cell, sometimes called the equator. Chromatids are attached by their kinetochores at the centromere by the spindle to the centrosome. This means that the sister chromatids are attached by their kinetochores, the dark red part on the picture, at the centromere, the light red part on the picture, by the spindle, the green lines spanning across the cell, to the centrosome, the two dark green circles. Anaphase. In anaphase, the microtubules extend from the centrosomes to the kinetochores during mitosis. The microtubules pull the kinetochores towards the poles of the cell. This gives a unique bracket shape to the nucleus, as the rest of the chromatid is dragged along when the pulling occurs. Sister chromatids are separated by the shortening of microtubules. This causes the pulling effect from both sides. Therefore, the distance between the chromatids increase as they are moved farther apart. The fourth stage is telophase. The nucleolus and nuclear envelope reform single centrosome per daughter cell. Now, each daughter cell has only one centrosome, which is replicated during the S phase on the next cell division cycle. Chromatids reach the poles and uncoil. The aftermath of mitosis. One centrosome is found in each daughter nucleus. One identical chromatid per daughter nucleus. Each chromatid contains an identical copy of DNA. Each nucleus is identical. The final stage of cell division is cytokinesis. This means the constriction of the cytoplasm to form identical cells. What exactly happens during cytokinesis? As the two cells start to form, the surface area increases between the cells. Therefore, new cell membrane must be constructed. In plant cells, however, they do not have centrosomes, nor a mitotic spindle, and after cell division, a new cell wall must also form, due to the increase in surface area. What is the importance of mitosis? First is immune response, then asexual reproduction, repair of tissue by cell replacement, replacement of damaged and dead cells, growth of multicellular organisms. Immune response. Mitosis is a process responsible for the cloning of P and T lymphocytes during an immune response in the body. Asexual reproduction. Mitosis is responsible for the production of new individuals of a species by a single parent organism. This is called asexual reproduction and is usually very rapid. Offspring are genetically identical to parents. Repair of tissue by cell replacement. Cell replacement is rapid in the skin and gut lining. Some animals regenerate whole parts, as starfish have the ability to regenerate one of their arms. Replacement of damaged and dead cells. We cannot say that cells are fixed, they are completely replaced. Cells constantly die and are being replaced by the identical cells, by the process of mitosis. The growth of organisms. Two daughter cells formed by mitosis have the same number of chromosomes. This makes them genetically identical, as they both have one copy of the DNA molecule. This allows the growth of multicellular organism from a zygote, from the first cell in a baby. Growth may occur over the whole body, as in animals, or confined to certain regions, as in plants. These regions are referred to as meristems. Thanks for watching.